Good evening, good evening. So welcome to the Macular Society Macular and Me webinars, for another one for this month. Last month's was on sort of emotional support and counselling, which went down really well. So if you attended that one, I hope you enjoyed it. But this one, we're back to our condition-specific sort of features, really. So the whole point of these was to talk about different conditions that weren't AMD on my stream. So for those of you who don't know me, my name is Colin Daniels, and I'm the Working Age and Young People Service Manager for the Macular Society. And we've been running these since last year. And we've got a whole back catalogue now of decent and really good, interesting webinars on our YouTube channel and on the Macular Society website. So if you want to have a look back at those things, then off you go. So this is a full webinar package. So as you may have noticed, you're unable to share your screen and you're already muted, which is great. But it doesn't mean that you can't get involved. So we do have a chat panel, as we all know these days about these things. So when our speaker, who I'll introduce in a second, is going through her presentation, if you want to ask any questions, just pop them in the chat, and then Jerry, who's here as well from the Macular Society, who I'll introduce as well in a second, she will just she'll read them out, and we'll all go from there. I think we're all getting quite good at this now. I mean, apart from me, who's stumbling over my words today for some reason. I don't really know why, but it is one of those things. I think it's because the clocks went forward. And that's all I should be in bed by now or something. I know I'm not quite there yet. Anyway, so without further ado, so we're going to introduce Jerry Hode first, who's the research manager for the Macular Society. Hi, Jerry. How are you today?
Great, thank you. Yep, I'm the research manager. I look after the research program, which is all the research projects that the Macular Society funds. We fund 7 or 8 new ones each year. Some of them are big grants of £250,000. Others are PhD studentships, which is this one. And welcome to Elena. And we joint fund her project with Retina UK. We like to collaborate with other charities where we can.
We absolutely do. And also, as you mentioned, we have a PhD student, Elena Piotter. How are you, Elena, today?
Oh, I'm great. Thank you for having me.
No, you're more than welcome.
Very excited to be here.
Yeah, we've had a few chats over the last couple of days, so it's been good, and so I'm really interested to hear what you've got to say. Now, I need to say that Elena is a research PhD student, so she has little medical knowledge. So and neither do we. But however, if you've got any questions around research and that sort of stuff, that's what we're here to answer today. Any medical information and things like that, obviously you need to either speak to your consultant or speak to them, the Macular Society Advice and Information Line, and they can point you in the right direction for that kind of medical support. All right, does that kind of clears that up? Am I right in saying that, Elena?
Yes, yeah, absolutely. Thank you so much.
Perfect, no problem. Okay, well, so I have Stargardt's disease, so we're here to talk about sort of research into treatments into Stargardt's. So that's what we're here to talk about today. So I'll just hand over to you, Elena. So if you'd like to share your screen, and I will put myself on mute and listen with intent.
Perfect. Can you guys, can everyone see my screen? Or can you guys see it?
Yep.
Cool, perfect. Cool, well, thank you so much, Colin, for that introduction, and thanks for having me. So as Colin mentioned, I'm going to be talking about gene editing for Stargardt disease. I'm currently a PhD student at the University of Oxford under Professor Robert McLaren, and as Jerry mentioned, I'm funded by Retina UK and the Macular Society, and I'm super, super grateful for that funding because it's been an absolutely amazing experience so far. So today I'm going to be talking about first a brief background about myself and how I ended up here. And then I'm going to talk a bit about like a background on science and the relevant science and research. To that kind of leads into the research that I'm doing. And so then, I'll delve more into the research that I'm currently conducting. So I'll get started. It's not going to, oh, there we go. Cool. So first things first, I grew up in Salt Lake City, Utah, and San Francisco, California. Very different places. One is quite mountainous, one is more coastal, but both are very beautiful and definitely recommend visiting. I attended the University of California, Davis while I was in California. And I majored in global disease biology and international relations, which are quite different from each other, but the university had a really interesting sort of presence because of their agriculture and veterinary background on gene editing and GMOs and stuff like that. So it was always kind of part of the discourse there. And yeah, they always like really talked about like how and when and why you should and shouldn't use this technology. And while I was there, I did research in a protein engineering lab where you could kind of modify reactions that proteins process to see if you could make it do what you want. And that's kind of when I realized like how you can harness things that exist in nature to kind of do more what you want to do, which is always just kind of blown my mind a little bit. And so because of that, I ended up doing a master's degree in the University of Groningen, which is in the north of the Netherlands, and did that in molecular biology. And for that, I did 2 research projects, one looking at like soil bacteria and the relationship with plants, which was it was very interesting, but not really what I wanted to do. And then the second ended up being in Professor McLaren's lab. And there I did a lot of research on, like different CRISPR-Cas systems, which I'll get into more later, and how they can be used for inherited retinal degenerations. And so going into that project, I didn't know that I wanted to do a PhD, and I didn't know anything about eyes, like nothing, let alone that retinal degenerations were even a thing. And it blew me away, the whole research program and everything. It was just amazing. And like the first week I was there, I got to see a gene therapy surgery that was happening, and it made me realize that the research is actually like very applicable and doesn't have some like super distant future that may or may not help society or whatever. And so I ended up starting to do a PhD there that began in October 2020 with Professor McLaren. And so, yeah, this research focuses on gene therapy for Stargardt disease. And so just some brief scientific background. I'll begin with what is a gene. So on the screen here, you can see a chromosome with this kind of stringy thing coming off of it, and that is unwound DNA. And so a gene can be any sort of region of this, varying in length from pretty small fragments to very large fragments. And it's a region of this genome that codes information in a combination of molecules. And the molecules that make this up are these here, the G, C, A, and T, so guanine, cytosine, adenine, and thymine. And so G always pairs with C and vice versa, and A always pairs with T and vice versa. And so this creates this code that can be read as information by cells. And the different sort of fragment lengths, are read in different ways. And so, so this can vary massively. So some genes can be up to about like 2 million bases, which is definitely on the larger end, but they can also be quite small. And so for this, you also have, for this like process, I guess, to happen, you have sort of 3 main steps that occur. So you begin with the DNA, and this then gets transcribed into RNA. So this is kind of like if you had a book and then it got compressed into, or not the book itself, but what you took from the book got compressed into a summary. And this shorter version is then what is used to create a protein. And so at any point though, throughout this process, something could go wrong. And often inherited genetic diseases come from a mutation in the DNA. Well, they do. And so this can then have a negative downstream effect where then the RNA is wrong and then the protein is either non-existent or misfolded, for example. And so it doesn't process the reaction that it should. And this then is what leads to disease or inherited diseases. And so this is the same with inherited retinal degenerations where you have faulty proteins, and this can then cause degeneration over time. And so for this, gene therapies that are in current use, one of the, actually I would say the main method that's been in use is the delivery of the correct gene to the relevant cells. And so the way this is done is a viral vector is used. So viruses have evolved, they're like super evolved to be able to find very specific cells. And typically they would in nature, they would deliver their own gene into a cell, and then kind of take over that cell, and the cell will produce more virus. And so this system was sort of altered in science, basically, where instead of the viral gene in the virus, you have the gene of choice that you put into the virus. And so the virus will still go to the same cell type, but instead of putting its own gene in there, it will put the gene that you want in there. And so this has been done with Leber's congenital amaurosis and Luxturna. Robert has done this with choroideremia and RP65. And so for this, the adeno-associated virus is used because it's really good at finding photoreceptor cells. And so, so it was quite revolutionary when this happened, only about 10 or 15 years ago. And so, it was very, very exciting. But like a lot of things, like a lot of things in science, there's still some shortcomings. But before I get into those, so the eye is also a really great target because it's readily accessible. You don't have to do any super invasive surgeries. You can also measure some very good clinical outcomes by seeing if people's vision is improving. So you don't actually, have to, extract tissue or anything. And that's also very non-invasive. And you also have a control. So you can inject one eye and then compare it to the other eye. So within one patient, you can tell whether the treatment is working, which is quite vital in figuring out whether these treatments are worth pursuing. So for Stargardt disease, the relevant gene is ABCA4, which is a fairly large gene. It's the most common gene that causes Stargardt disease. So if you have a mutation in this in both alleles, then you end up having Stargardt disease. And so one of the main issues that current viral vectors face, or the AAV, the adeno-associated viral vector that is the common gene therapy vector, is that the capsid in which the gene is delivered is too small. So it can only deliver fairly small genes. And unfortunately, ABCA4 is too big to fit into it. So this method doesn't work. So what you have here on the screen is a very large cat trying to fit into a very small box, which is probably the best analogy I could think of. And so there are a number of things that have been tried to sort of get around this issue. So first, of course, you try to get a bigger box where other viral particles have been tried. So lentiviral vectors have been tested. And you could, like artificial synthetic particles have been created. So these are called nanoparticles, and these have been made out of lipids or various like metals and stuff like that to try and deliver to the retina. And then it's also been tried to be split. So for example, this dual vector delivery system, part of the gene is in one viral particle and part of the gene is in a separate viral particle. And then both of these are delivered to the relevant cells. And once the genes are injected in the cell, the gene will recombine. And so this has been tried with both DNA and with proteins. And so a lot of things have been tried. An alternative method that has kind of come to light in the last also 10 to 15 years is CRISPR-Cas. And so that's been in the news a lot because 2 years ago, I think, the people who kind of discovered this editing system won the Nobel Prize, Jennifer Doudna and Emmanuel Charpentier. It's an incredible thing they discovered. And so this stems from a bacterial immune response to viral attack, actually. And so this is kind of how it evolved. So CRISPR is a mouthful. It stands for Clustered Regularly Interspaced Short Palindromic Repeat. I don't know why there's so many long acronyms and words for something or for things in science, but that's the way it is. So what you have on this screen here is this sort of red diamond and a large black circle with red squigglies inside. The red diamond represents the virus and the black circle represents the bacteria. This is my basic art that I'm capable of. And so the virus injects these red squigglies, which represent its DNA. And so similar to with cells, this would sort of hijack the bacteria and make it produce more virus. And so in response to this, the bacteria has evolved this system called the Cas enzyme, the CRISPR associated system, that can identify these foreign DNA particles and chop them up basically. But then it's evolved another step where it takes those small pieces and integrates it into its own genome. And this is then sort of like a repertoire of a memory where it can then remember in the future if it gets attacked again, I've had this happen before, and it will find those similar sequences and chop those up much faster. And so it's pretty amazing how this evolved. So then Jennifer Doudna and Emmanuel Charpentier have been researching this for a while, and other scientists, I'm sure, thought maybe this could be used to edit cells. And kind of accidentally, it was realized that these small sequences that it uses to recognize a virus, they can actually be any sequence. They don't necessarily need to just be a viral sequence. So for this, here's another, a few other keywords. Oh, sorry, before going on. But yeah, so basically the system could then be used to target really anywhere in the genome, and then you could edit it. And so going forward, there's a few more keywords. So there's Cas9s, which are used to target DNA. There's also Cas13s, which are used to target RNA. And these Cases can come from different bacterial species, and because of that, they also vary in size. So they also have a gene that allows for the enzyme to be produced. And yeah, so that gene can vary in size, which becomes important down the line. And one other word I guess I'll be throwing around is this thing called a PAM site. And so not only when a Cas is being guided to a target, does it need this guide sequence, it also needs a smaller species-specific sequence right next to it. So it doesn't necessarily, it can limit like what you can target because yeah, it needs to have both this PAM sequence right next to the target. So yeah, I'll explain that in the next couple slides on how it works. So here you have a Cas9, which is this cloud-shaped thing in the center of the screen. And then you have a guide RNA, which is this red squiggle drawn here. And then you have a ladder across the screen that represents the DNA. And so what happens is the Cas9 and this guide RNA combine. And then the Cas9 will search for the guide sequence within the DNA, as well as the PAM site. And so once it has found this match, it will bind, and then it unwinds, and there's a lightning bolt that just appeared. And that means that is to indicate that the Cas9 then cuts there. And so then this will render the gene inactive. And so this is sort of the system that was first developed with CRISPR-Cas. And so this was super exciting because you could test, the effects of certain genes, what would happen if they were rendered inactive. There were also other instances, where mice infected with HIV were targeted with Cas, and it significantly reduced the amount of HIV infection, stuff like that. So it had an amazing prospect in the scientific community. More recently, this has been vastly optimized. So this new system that I work with is called base editing. So here you have a picture of a gloved hand with tweezers changing out a single base in DNA. And I wish it were that easy, but unfortunately it is not. But it does work quite like that in the sense that, I'll get to that in a second actually. So just to reiterate these necessary components. So you have in this process, so you have the DNA getting transcribed to RNA, and then that gets translated into a protein. And so you could target, using this base editing system, you can target the DNA and the RNA, which is pretty amazing. And you could correct a mutation there. And then this would have a downstream effect to create the desired protein. And so the way DNA base editing works, I'll start with that, is similar to what I previously explained with how Cas9 cuts DNA. In this case, the Cas9 still combines with the guide RNA and searches for both the PAM sequence and the adjacent guide and binds to the DNA. When the DNA unwinds, though, instead of cutting, you have there's another third component that is fused to the Cas9. And this enzyme then processes a reaction that can make a base change. So it could go from an A to a G or from a T to a C, depending on what that enzyme is. And so while that seems quite limited, it does actually address a huge number of mutations, which I have later on in the slides. So then once that is corrected in the DNA, then in theory, all of the downstream RNA and all of the downstream protein would be corrected and functional. So some of the main things to consider with this, though, is that, if this sequence if there's a very similar sequence elsewhere in the genome, it could accidentally bind there and cut or edit there. And so then while you're correcting some mutations, you might be introducing others. The other is that, you do have this PAM sequence, which is quite specific. So it kind of reduces the number of mutations you can target. So these are kind of just things we consider and are trying to optimize to make the best sort of base editor system. RNA base editing works very similarly, except that it targets RNA. So you have this Cas13, which is again represented by this like cloud-like structure. And it too combines with the guide RNA. It finds the target, and then you have this other fused enzyme that mediates that base change from an A to a G, for example. In this case, it can only do A to G, actually, very efficiently. Luckily, this system does not require a PAM site, so basically any mRNA can be targeted, which is awesome. The only thing with this is because it's downstream of the DNA, you do need, it's sort of hypothesized that you probably need a higher editing efficiency for this to be effective, especially because RNA does naturally degrade over time, so it constantly needs to be working. That being said, the degradation of the RNA is a plus, because if there is an off-target effect, for example, it doesn't have any implications really, because it's not a permanent change, or it has significantly reduced implications compared to if you made an irreversible change in the DNA. And so these are kind of the things we consider when using these systems, is like both, of course, safety and efficacy. So then this in gene therapy, what would this look like? And it would be very, very similar to how all other gene therapy works, where you would use a viral vector to deliver the Cas gene plus the guide to a cell, and then the cell would produce that, and then it would go and perform editing in the cell. And then, so then some things to consider in this process, especially on the note of delivery to the cells, is that the size of the Cas gene does matter quite a bit, similar to with ABCA4. A lot of Cas species can be quite large and might not necessarily fit into the preferred adeno-associated viral vector. But the nice part is there's a lot of bacteria out there, so there's a lot of new species being discovered. There's a lot of ways to make these smaller. And so this is constantly being optimized. Likewise with the PAM sequences, these have been, even in the last year, the flexibility on this front has massively increased. And so that is much less a limitation. The efficiency has also been massively improved, as has on-target editing, while reducing off-target editing. And then one of the other main things that is still we're quite in the dark about is the length of the efficacy and whether you would have to re-administer treatment. Because this has obviously not really been tested in people, we don't know how long it will last for, and the length of time that would be good for. And so for people, I think for pathogenic mutations and genetic disease, this is a pretty incredible system. when you look at the total pathogenic human mutations, about 63% of them are single base mutations that are G to A and T to C, which could be corrected by these systems. So that is just absolutely amazing. I think there's either 32 or 37,000 pathogenic mutations in the genome that we know of. So that would address quite a high number of those. And so in ABCA4 as well, in Stargardt, there's about 1,200 known pathogenic mutations that can result in Stargardt or other ABCA4 associated retinopathies. And so 63% of these as well are targetable by these two systems. So that is just like absolutely amazing, and does, I think, attest to the relevance of the technology for addressing ABCA4 mutations. And CRISPR is evolving super, super quickly and just improving constantly. Like, it's so hard to keep up with all the publications that are constantly coming out on this. And so it is just amazing to see, where this field will be in 10 years and see it hopefully, help with, provide a solution for something for Stargardt or any ABCA4 associated retinopathies. So yeah, it's just been an exciting project so far, and I'm really, really excited to see where it goes. Here's a picture of my lab. Thank you so much for having me. I look forward to the questions, and thank you very much.
No, thank you, Elena. That was really interesting. It's just things move so quickly, don't they? It's incredible.
Yeah, it's hard to keep up sometimes.
Yeah,
most of the time.
Yeah, it's crazy. It's crazy. It's it does believe. I said this to you the other day. It does look like there's certainly some kind of light. It might be a long way in the distance. Some light in the distance. Finally. So Jerry, has there been any questions come up?
Yeah, we've got one so far asking how long until this goes to clinical trial.
Oh, okay. That's a really hard question.
Tricky one.
I don't think there's an answer to that yet, unfortunately. I think, just with like approval processes and stuff like that, it takes so long. And yeah, this has to be tested in cells first, and then it's quite a while, I think. But it is exciting at how quickly the field is moving. So yeah. I wish I could give a more definitive answer.
No, I don't blame you. It's how long is a piece of string? These research takes a long time, and what you're doing at the moment is laboratory-based research. And then once you've proven that it can work in the lab, then they have to do a lot of work before they can take it to clinical trial as well.
Yeah, and the phases
to be as sure as they can that it's safe to give to a human being.
Of course, yeah.
Yeah, it's frustrating. I know how long these things take, but the progress is really great.
Yeah.
I've got a question of my own, actually, while we're waiting for any more questions to come in. The CAS systems, where do you get them from? Do you have to extract them from the bacteria, or are they made artificially?
So I actually just order them from there. you can order them in a bottle, like different websites. I think they're just extracted from bacteria. I actually don't know where they originally get them from, like how they just target those. I probably should know that. But yeah, there is like because I guess there's a ton that come out every year, new CAS species and stuff from a number of different bacteria. But there's some that are just like far better than others in terms of their efficiency and their on-target editing. The two that we work with are the Staph aureus CAS and SP CAS mostly, because they just work so much better and are just very reliable. And especially this Staph aureus, given its small size, has been awesome. So I think like we've mostly been sticking with those so far. But yeah.
Right. Okay, another one's come in from Catherine. When it does go to trial, how would someone get involved as a research participant? So how would they be able to join the clinical trial?
Ooh, I don't know.
I might be able to help with that one if you're not sure.
Yeah, go for it.
Okay, I think increasingly to be eligible for a clinical trial, you probably have had to add a genetic diagnosis. It certainly will be a big step forwards to be able to, because the clinicians will want to know what the mutation is in your ABCA4 gene, so that they can decide whether you might be eligible for the trial. So I always encourage people when I'm talking to them to, if they don't have a genetic diagnosis and they're interested in joining clinical trials, that would be a good first step. Otherwise, you just talk to your ophthalmologist, let them know that you're interested in research, and then they'll consider you if there's a trial running at your hospital. Or keep in touch with the Macular Society. We advertise clinical trials taking place and recruiting. So we're often asked to help to recruit with clinical trials. And you might hear about it at one of our conferences or through our website, a news item. So it's really staying in touch with what's going on. And then you'll hear about clinical trials. And, you know, if your doctors know that you're interested, they'll let you know if they think you might be suitable for it.
Thank you.
Right. Next one, sorry. Scroll down a bit. Have you got a virus that is big enough to be a delivery vector?
Yeah, so there is the lentiviral vector, and that has been used. I think it does, if I remember correctly, I think it has issues targeting retinas, like the retina. So it might not necessarily, like it can be used for gene therapy, but I don't think it's particularly efficient for retinal degenerations, unfortunately. It's a huge virus. I mean, the AAV can package about 4.7 kilobases of DNA, which is not a lot, it's quite small. And the lentiviral vector can package up to, I think, about 18 or 20 kb. So it's about 5 times as big, which, given how small everything is, can be quite difficult to get it in there.
How would a treatment like this be administered?
It's a subretinal injection is how they do the AAV deliveries. So I'm not exactly sure how, like the ins and outs of it. But yeah, so they produce the AAV, and the way my professor has explained it is they do an injection to make a bleb. So it's kind of like a bubble, and then that they can do like an injection immediately after that in the same space. And so then that will like get the virus to the affected area, is how I understand it. I'm sure there's a much more technical medical explanation for it than what I can give.
Sounds perfect to me, to be honest. Since my little bonds, that does cost.
And
Elena, is that what you saw in theatre, Robert McLaren in theatre? Is that what he was doing?
Yeah, so I can't remember what gene therapy he was doing because he has a number of different clinical trials going. But yeah, that's kind of what they do. Yeah, it was kind of crazy, yeah, because they take like a speculum to like pop out the eye a bit so they can access it better. But you don't actually see a lot because a lot of it happens like he does it with a microscope and stuff, and it's all very tiny. And so, yeah, it was very cool though.
Great. I'll ask this one, but I may have to help you with this one. I don't know whether you know. It says, how can someone request genetic testing? My local hospital said there's no treatment, so we are discharging you.
Oh, wow.
Do you want me to help with that one? I got.
Yeah, I think Retina UK has a pretty good. Oh gosh, I don't want to say that though, and then have that not be right.
No, you're right. They do have a very good sort of secondary website, and I can't remember the name of it, which explains all about genetic testing.
It's called
basically, there was something a few years ago called the 100,000 Genome Project, which tested 100,000. It was about 70,000 people were involved. They either had rare diseases like Stargardt's, or they had cancer, and they were looking to sequence all the genes of about 70,000 people and 100,000 genomes. There was a disparity between the two because with the cancer patients, they took the cancer gene, cancer cells, and the normal cells and sequenced both. So from that 100,000 Genome Project, they now have developed that into the NHS Genomic Medicine Service. So the idea is that facilitate across the whole of the NHS genetic diagnoses where that's appropriate. So it has therefore become a lot easier to get a genetic diagnosis than it was certainly 10, 20 years ago. So it's really a question of going back to your hospital and asking whether you can have a genetic diagnosis. I don't know what you have, Catherine. If you want to pop it in the function, if you have Stargardt's disease, you know, there may be issues questions that you want to ask about inheritance. Generally speaking, if you have a genetic diagnosis, you will have a discussion with a genetic counsellor before the test is done, so that you fully understand the implications of the test and what it might be able to tell you. But there is a lot of information online that we can point you to, or you may like to listen to the talks from the 2021 Macular Society conference, where we had a couple of genetic counsellors talking about exactly this, and that's exactly
Jerry, or you could join our session next month with Andrew Webster and a gene counsellor who will explain the whole process.
Brilliant, aren't we? Doesn't that feel smooth?
It does.
Yeah.
Okay, so Catherine's got Stargardt's. Okay, so I would recommend, there's a lot of information online about genetic testing. If in doubt, we can post up some links. So let me.
And I think the website you were referring to was set up by Maria Musaji as well, wasn't it? So that's right. Yeah. Gene Vision, it's called.
Okay, so let me find. Next one is, I'm being sent to a geneticist before I've actually seen an ophthalmologist. It's based on my OCT images. Seems an odd way round. Yes, I kind of agree.
Mm-hmm.
So they're thinking that the genetic test will help with the diagnosis, I'm assuming.
Yeah. It still takes quite a long time for those genetic results to come back. I mean, it's a lot quicker than it was. I think mine came back in about 10 weeks.
Oh, you've had one, I didn't realise. Yes, and not everybody does get a diagnosis, but the diagnostic rates are improving all the time with everybody that they test, and they to discover what the mutation is that's causing the problem. They learn it a bit more. So it is a growing area of medicine, really, genetic testing. Right. Becky says, yes, based on my age, they've said it must be genetic. Yes, sometimes people come to us at the Macular Society saying, oh, I've been told I've got macular degeneration, and they're maybe in their 30s, and they may think sometimes they think it's age-related macular degeneration, and we say, no, you're too young. It's probably a macular dystrophy like Stargardt's or best.
Yeah, there's this professor in New York who, in one of his lectures, like shows images of the retinas for all the different macular dystrophies, and they look identical to me. Like, I don't know how you would be able to diagnose just on the images. I'm just like amazed at ophthalmologists that they can like distinguish that, and then obviously like need the genetic confirmation, but it's just it's crazy. I was like, oh my gosh. It's like identical, but yeah.
Yeah, but I think they're able to define the diseases so much better than they were in the past.
Yeah.
You know, in the past, they were just looking at the back of the retina and sort of interpreting what they could see.
Yeah.
And some diseases look quite similar, although they've got a completely different genetic basis. So now the genetic testing with the OCT scans, they're much more defined in the diagnosis, which is great.
Yeah.
Which it does mean some of the names are changing a bit. Or not as applicable as they used to be. So Gail's asking, I'm in Northern Ireland, are the trials England-based only? Quite often they can be, but certainly we know of clinical trials going on elsewhere, all over the UK. And this is one I'm not quite so sure about. Any news on dome macula, which I have heard of?
Ooh, on what? Sorry. What?
Dome macula.
Ooh, I have no idea.
Not off the top of my head, I'm afraid. We can follow that one up for you.
Yeah.
I certainly haven't heard of any clinical trials on it.
Yeah, drop me an email and at colin.daniels@macularsociety.org, and we'll have a little look. Okay.
And the next one from Greg, as I understand it, everyone with Stargardt's has a different mutation in either copy of the gene. So for treatment, would the injection have to be individually created, or would it be off the shelf? question.
So in this case, it would, that's one of the main, I think, issues that the base editing system would have, is because there's so many different possible mutations for Stargardt. And like some definitely are more common than others. You would have to create a guide sequence on an individual basis. Luckily, because the Cas, for example, is quite separate from the guide, it's not like one giant gene sequence. You could still do that as a dual vector. So you'd only have to, like, you could still, like, you'd only have to like make the one vector uniquely for that person. So yeah, I guess that's kind of a logistical thing that would have to get figured out, but that would be way down the line. So I guess we just want to see if it works first. But yeah, normally with the gene therapy, like if there were the potential to make a delivery vector like large enough, you could just put a fully functioning, like a full correct sequence for ABCA4 in that and deliver that, and that would work for everyone in theory. I think there are rare instances where there's like dominantly inherited ABCA4 and stuff like this where it might not necessarily work, but I think for the vast majority of patients, it should work in theory.
Next one, how are you identifying or obtaining data for your research projects? Is it only dependent on participants, or are you accessing an existing database?
The computational stuff that I've done, because we wanted to see the relevance of this in terms of how many mutations could be targeted by it, given this PAM site relevant, like sort of in, oh my gosh, this consideration, I guess. So for that, I use, there's a number of different databases you can look at. I think ClinVar, and then Nomad, and LOVD, which is the Leiden Open Variation Database. Those are kind of the three main mutation databases for any gene. Yeah, and so there you can see all of the different published and like entered into the database variants. And so I use that for those. For the lab work, yeah, that is just, yeah, kind of using existing Cas systems to target different mutations, and then hoping that they get fixed. So then we send those samples for sequencing and see if they have been corrected. So yeah, does that answer your question?
We'll see. So the next one is somebody listening from Ontario, Canada.
Yeah.
Wonders of Zoom.
Yeah.
They work with geneticists at something called Sick Kids Toronto. That's great. Do you share or look for candidates for clinical trials worldwide?
Ooh, I'm actually not certain.
Do you want some help?
Yeah.
It depends.
Yeah, sorry, go ahead.
Yeah, if it's a large trial, it will often take place in lots of different countries. Probably mostly America, and then second sort of on the list of European countries. But yes, lots of trials are worldwide. Don't hear about Canada a great deal. It's usually America and Europe. And then the second question is, this is one for Colin, and your seminars are new to me. Where can I find the back catalogue?
If you just go onto the, if you Google, actually, I found it by accident earlier today, actually, if you just Google Macular Society webinars, the top two will be registering, and the next one below on Google will be watch back. Or the other way to do it is to go onto YouTube, find the Macular Society and subscribe to our channel, because every time a new video of these webinars gets done, it'll be popped up, and all our old ones are there as well. Or the old ones, the previous ones is probably a politer way of saying that. I mean, there's a good catalogue. I mean, we did, I think on the AMD stream, there was 11 or 12 last year, and on this stream there was I think I did 13 in total. So there's plenty to watch back, all about different subjects. So it's very it's a good resource, and it's growing every day, which is fab.
Great, thank you. So one from Kathleen. I've been told that while I have familial dominant drusen diagnosed 40 years ago, it's not genetic. Is it early onset AMD?
I have no
I'm
idea.
not sure.
No idea.
I'd have to look into that one.
Yeah, that's definitely one for your your consultant, I think, or the trip to the eye clinic.
There is something called early onset AMD. We've just finished a project with Graham Black at Manchester on early onset AMD.
Yeah.
And
hopefully,
that definitely is genetic.
Yeah. And hopefully I'll be doing a webinar in June on it. It's sort of in, I think. But watch this space. That's what I would say.
Okay, so she's now put. They told me it was early onset AMD, so that is a form of what we call collectively the macular dystrophy. So the genetic conditions that cause macular disease that affect younger people. So do look out for. Yes, Graham Black's just finished a research project funded by us on early onset AMD. And there'll be information on our website. If you go onto the research section and explore our projects, there's a web page about that project, Graham Black in Manchester. And if you're a member of the Macular Society and you get the Side View magazine, there will be a feature on it, or there has been a feature on it, I think, very recently in one of our magazines.
Excellent. Excellent. And this would be a good opportunity to say, and if you're not a member, you could take, we do a 6-month free, trial. So you just join for free and read the magazine. See how beautifully, it was seamless again. I dropped that in nicely.
We're such a team, yeah. Yes, I mean, you get the latest version of Side View, but I think you might also get access to the website where the old versions are.
You do. And it's only £22 after that first 6 months, he says, just dropping it in there as well. Have we got, what a bargain. Yes. Have we got any more questions in the chat? No,
that's the last one we've got coming through.
Perfect. Brilliant. Excellent. Well, I know we've got a bit more time. So, Jerry, do you want to, so we've just had our round of research. We're sort of, what's happening with our research stuff at the moment?
Okay, so we spend about £1.5 million every year on funding new projects. And as I said at the beginning, that allows us to fund 7 or 8 new projects a year. It varies because they all have different amounts that they cost. So we fund 3 types. We fund big research grants, PhD studentships, and then a relatively new form of grant, which is the seed corn grant, which is just a 1-year small project designed to allow researchers to generate data, to look at something really innovative, and then that data can be used to support a larger grant application. So there's information about all those types of projects on our website. And we always make public our research, and we're often doing webinars and things, so people can see the results. And we liaise really closely with our researchers, and they send us in regular reports on how the project is going, and then a final report at the end. And we have a research committee full of ophthalmologists and top optometrists and some of our trustees as well. And they're the ones that choose the projects for us. So we have experts choosing which projects are the best to fund. So they have to be really good scientific quality, value for money, and from excellent researchers who've got experience in that area. And we love our PhD students as well, because we want more people to come into this area and do more research. We need those new young researchers through into their new careers.
So I'm giggling because my guide dog is Woody is being a mischief. He's trying to chuck a soft toy at my housemate. Anyway, so well, that's really good. So funny. Anyway, sorry, I got distracted. I do apologize. Elena, do you want to stop sharing your screen so we can, yes. Please and thank you. Excellent. Well, I think I don't think we've got any more questions, so we've kind of sort of drawn to a close, I think, which is fine, which is excellent. So brilliant. So we've got a few thank yous coming in, which is nice. So anyway, Elena, thank you ever so much for doing that. I mean, you explained everything in such an easy way, and I mean, I'm a bear of very little brain, and I understood it. So if I did, I think everyone else did as well. It's been really informative, and as I say, it does show that things are definitely moving in the right direction, and we're understanding a little bit more about genetics and gene sequencing and gene therapies, and hopefully that will eventually help sort of solve some of the problems that people are having with their inherited retinal diseases. So thank you very much for your time this evening.
Thank you so much. It was really nice.
Great.
Thank you.
Excellent. And Jerry, thank you as usual.
You're welcome.
But marvelous. As I slowly slid in earlier, the next one almost seamlessly is with Professor Andrew Webster from Moorfields, and he's going to talk about why we should understand more about our genetics and our genes. And he's invited a gene counselor along to explain what they then do with that information. I went right north at them for a second. What they then do with that information. Anyway, so it leaves me to say thank you very much for all attending. If you want to watch this back, it will be on the website and on YouTube in the next week or so. There will be in your inbox, as soon as I end this session, a little survey just about information, just to tell us how you found this. Please fill it in and send it back, because we can only make these better with your input. So thank you very much for coming, and we will hopefully we'll see you all next month. So thanks very much. Bye-bye.